Welcome to Chapter 2 of Class 11 Biology, Biological Classification. In this fascinating chapter, we'll explore how scientists have organized living organisms into classification systems that reflect their relationships and characteristics. Since the dawn of civilization, humans have attempted to classify living organisms, initially based on practical needs for food, shelter, and clothing. Aristotle was the first to attempt a more scientific basis for classification, using simple morphological characters to classify plants into trees, shrubs, and herbs, and dividing animals into those with and without red blood. Linnaeus later developed the two-kingdom system with Plantae and Animalia kingdoms. However, these early systems had significant limitations. They didn't distinguish between eukaryotes and prokaryotes, unicellular and multicellular organisms, or photosynthetic and non-photosynthetic organisms. This led to the development of more comprehensive systems, culminating in Whittaker's five-kingdom classification that we'll explore in detail. Let's dive into R.H. Whittaker's five-kingdom classification proposed in 1969. This revolutionary system addressed the limitations of earlier classifications by using multiple criteria including cell structure, body organization, mode of nutrition, reproduction methods, and phylogenetic relationships. The five kingdoms are Monera for prokaryotic organisms, Protista for unicellular eukaryotes, Fungi for heterotrophic organisms with chitinous cell walls, Plantae for eukaryotic photosynthetic organisms, and Animalia for heterotrophic multicellular organisms without cell walls. This comprehensive approach was a significant improvement over the two-kingdom system, which didn't distinguish between fundamental differences in cellular organization and mixed together organisms with vastly different characteristics. The five-kingdom system provided a more accurate reflection of evolutionary relationships and biological diversity. Now let's examine the key characteristics that distinguish the five kingdoms. Looking at the comparative table, we see that only Monera has prokaryotic cells, while all other kingdoms are eukaryotic. Cell wall composition varies significantly. Monera has non-cellulosic walls, fungi have chitin, Plantae have cellulose, and Animalia lack cell walls entirely. Nuclear membranes are absent only in Monera. Body organization progresses from simple cellular structure in Monera and Protista to more complex tissue and organ systems in Plantae and Animalia. Nutrition modes also show clear patterns. Only Plantae are exclusively autotrophic, while only Animalia and fungi are exclusively heterotrophic. Monera and Protista show both autotrophic and heterotrophic nutrition. These distinguishing characteristics form the basis for separating organisms into different kingdoms and reflect fundamental biological differences. Let's explore Kingdom Monera, which consists entirely of bacteria, the most abundant microorganisms on Earth. Bacteria are found almost everywhere, from everyday environments like soil to extreme habitats like hot springs, deserts, snow, and deep oceans where few other life forms can survive. Many bacteria live as parasites on other organisms, while others form beneficial relationships. Bacteria show the most extensive metabolic diversity among all organisms. They are classified into four morphological types based on shape: spherical cocci, rod-shaped bacilli, comma-shaped vibrio, and spiral spirilla. This shape-based classification helps in preliminary identification. Bacteria can be autotrophic, synthesizing their own food through photosynthesis or chemosynthesis, or heterotrophic, depending on other organisms or dead organic matter. This metabolic versatility contributes to their success in diverse environments. Now let's examine the two major groups within Kingdom Monera: Archaebacteria and Eubacteria. Archaebacteria are special bacteria that thrive in extreme habitats where few other organisms can survive. They include halophils in extremely salty areas, thermoacidophiles in hot springs, and methanogens in marshy areas and the guts of ruminant animals. Archaebacteria differ from other bacteria in their cell wall structure, which enables survival in harsh conditions. Methanogens are particularly important as they produce methane gas from animal dung, contributing to biogas production. Eubacteria, or true bacteria, include thousands of species characterized by rigid cell walls and flagella for motility when present. Cyanobacteria, also called blue-green algae, are important eubacteria that have chlorophyll like green plants and perform photosynthesis. They can be unicellular, colonial, or filamentous, and some can fix atmospheric nitrogen in specialized cells called heterocysts. Let's discuss bacterial reproduction and the unique characteristics of Mycoplasma. Bacteria reproduce mainly by binary fission, a simple asexual process where one cell divides into two identical cells. Under unfavorable conditions, many bacteria produce spores that can survive harsh environments for extended periods. Bacteria also exhibit a primitive type of DNA transfer that resembles sexual reproduction, though it doesn't involve true gamete formation. This genetic exchange occurs through transformation, transduction, or conjugation and increases genetic diversity, allowing bacteria to adapt quickly to changing environments and develop antibiotic resistance. Mycoplasma are remarkable bacteria that completely lack cell walls, making them the smallest known living cells. They can survive without oxygen, and many species are pathogenic in animals and plants. Their lack of cell walls makes them resistant to antibiotics that target cell wall synthesis, presenting challenges for treatment. Now let's explore kingdom Protista, which includes all single-celled eukaryotes. The boundaries of this kingdom are not well defined, and it serves as a link between plants, animals, and fungi. Protists are primarily aquatic and show tremendous diversity. Being eukaryotes, they have well-defined nuclei and membrane-bound organelles. Some have flagella or cilia for movement, and they reproduce both asexually and sexually. Protista includes several groups: Chrysophytes, diatoms and golden algae, Dinoflagellates, Euglenoids, slime molds, and Protozoans. This kingdom represents a catch-all category for eukaryotic organisms that don't fit neatly into other kingdoms, reflecting the transitional nature of many protists. As our understanding of evolutionary relationships improves, the classification of protists continues to evolve, illustrating that classification systems are human constructs that reflect our current biological knowledge. Let's examine two important groups of photosynthetic protists, Chrysophytes and Dinoflagellates. Chrysophytes include diatoms and golden algae, desmids, found in both freshwater and marine environments. They float passively as plankton and most are photosynthetic, playing crucial roles as primary producers in aquatic ecosystems. Diatoms have unique cell walls with two overlapping shells embedded with silica, making them virtually indestructible. Over billions of years, diatom cell wall accumulations form diatomaceous earth, used in polishing and filtration. Dinoflagellates are mostly marine, photosynthetic organisms that appear in various colors depending on their pigments. They have stiff cellulose plates in their cell walls and typically two flagella in perpendicular arrangement. Some dinoflagellates, like Gonyolics, can undergo rapid multiplication causing red tides that color the sea red. Toxins released during red tides can kill marine animals, demonstrating how microbial population explosions can impact entire ecosystems. Now let's explore euglenoids and slime molds, two unique protist groups. Euglenoids are mostly freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich pellicle that makes their body flexible. They have two flagella and exhibit both plant-like and animal-like characteristics. In sunlight, they are photosynthetic with pigments identical to higher plants, but when deprived of sunlight, they behave like heterotrophs, predating on smaller organisms. This dual nature makes them fascinating examples of evolutionary transitions. Slime molds are saprophytic protists with remarkable life cycles. Their bodies move along decaying material, engulfing organic matter. Under suitable conditions, they form plasmodium aggregations that can grow over several feet. During unfavorable conditions, the plasmodium differentiates into fruiting bodies with highly resistant spores that can survive for years. Their life cycle alternation between amoeboid and spore-forming stages demonstrates remarkable adaptability to changing environments. Let's examine protozoans, the animal-like protists. All protozoans are heterotrophs living as predators or parasites, and they're considered primitive relatives of animals. They're classified into four major groups based on locomotory structures. Amoeboid protozoans move using pseudopodia, false feet, and live in various aquatic environments. Some, like antamoeba, are parasites causing diseases like amoebic dysentery. Flagellated protozoans use flagella for movement and include both free-living and parasitic forms. Trypanosoma causes sleeping sickness. Ciliated protozoans are aquatic organisms with thousands of cilia for movement and feeding. Paramecium is a classic example with a specialized gullet for food intake. Sporozoans lack locomotory structures and have infectious spore-like stages in their life cycles. Plasmodium, the malarial parasite, is the most notorious sporozoan, causing malaria with devastating effects on human populations. Each group demonstrates different evolutionary adaptations for survival and reproduction. Now let's explore kingdom fungi, a unique kingdom of heterotrophic organisms showing great diversity in morphology and habitat. From the mushrooms we eat to the mold on bread, fungi play crucial roles in ecosystems and human affairs. They're cosmopolitan organisms found in air, water, soil, and on animals and plants, preferring warm, humid environments. Most fungi are filamentous, with bodies composed of thread-like hyphae that form networks called mycelium. Some hyphae are coenocytic tubes filled with multinucleated cytoplasm, while others have septae or cross walls. Fungal cell walls contain chitin and polysaccharides. Most fungi are heterotrophic, absorbing soluble organic matter from dead substrates as saprophytes, or from living plants and animals as parasites. They can also live as symbionts in lichens, with algae, or as mycorrhiza, with plant roots. Fungi store food as glycogen, similar to animals, highlighting their distinct evolutionary pathway. Let's examine fungal reproduction and classification. Fungi reproduce through various methods: vegetatively by fragmentation, fission, or budding; asexually by spores like conidia, sporangiospores, or zoospores; and sexually by oospores, ascospores, or basidiospores. Sexual reproduction involves three steps: plasmogamy, fusion of protoplasms; karyogamy, fusion of nuclei; and meiosis in the zygote resulting in haploid spores. In some fungi, an intervening dikaryotic stage occurs with 2 nuclei per cell. Fungi are classified into 4 main classes. Phycomycetes include aquatic fungi and plant parasites with a septate, coenocytic mycelium. Ascomycetes, or sac fungi, have septate mycelium and produce ascospores in sac-like asci. Examples include penicillium and yeast. Basidiomycetes, or club fungi, include mushrooms and produce basidiospores on basidia. Deuteromycetes are imperfect fungi, where only asexual reproduction is observed. This classification reflects differences in mycelium structure and reproductive strategies. Finally, let's explore viruses, viroids, prions, and lichens, organisms not included in the five-kingdom system. Viruses challenge our definition of life, as they have inert crystalline structures outside cells but take over host cell machinery to replicate. They consist of genetic material, DNA or RNA, surrounded by a protein coat called capsid, with no virus containing both DNA and RNA. Plant viruses usually have single-stranded RNA, animal viruses have various nucleic acid types, and bacteriophages usually have double-stranded DNA. Viroids, discovered by T.O. Diener, are even smaller than viruses and consist only of RNA without protein coats. Prions are infectious proteins causing neurological diseases like mad cow disease, BSE, and Creutzfeldt-Jakob disease in humans. Lichens are symbiotic associations between algae, phycobiont, and fungi, mycobiont, where the algae provide food through photosynthesis and the fungi provide shelter and absorb nutrients. Lichens are excellent pollution indicators as they don't grow in polluted areas.